Hey everybody, this is Rayon with Oceans for Everyone. So in this video, I'm going to try and go quickly. We're going to talk about CTD profiling and Niskin bottle sampling, and it's all about understanding the different characteristics of the water body, the physical, chemical, biological characteristics, right? CTD stands for conductivity, temperature, depth, right? So what is CTD profiling? So it measures the conductivity, temperature and depth, and other things if you want to add like oxygen or chlorophyll concentration. And so what we do is we collect continuous profiles of the water body. That is its whole purpose. And it's all for understanding water mass distribution, and all kinds of other things like physical and chemical properties. All right, so the components of a CTD instrument. Okay, so there's a conductivity sensor that measures salinity through electrical conductivity of the water. Then there's a temperature sensor, and then there's a pressure sensor, which is really important because that converts the pressure data to depth. All right, you can also do like turbidity and pH and all kinds of other stuff. All right, so the CTD profiling procedure. All right, so you have to prepare the instrument. You have to calibrate the CTD, and we do that usually just by leaving it at the water surface, submerged just below the surface for like a minute or two. And then when it's ready to go, you lower the CTD through the water column, and you can collect real-time data as it descends. All right, and then you retrieve the CTD and you download the data. And then once you have the data, you do the analysis and you can plot profiles of temperature and salinity and all that other stuff. So Niskin bottle sampling. So the Niskin bottle is just a cylindrical water sampler designed to collect water at specific depths. And applications are like, again, this chemical, biological, even microbiological studies. So the components of a Niskin bottle are like the cylindrical body, usually, and you have it's made of PVC or plastic or something resistant to seawater, right? And then you have the end caps that automatically seal when they're triggered at the desired depth. Whenever you reach the depth you want, you can trigger them either using a remote trigger or by messenger or electronically. Now, a messenger is like a little thing, a little piece of iron, usually like bronze, I think, or lots of the times. You send it down the wire, it trips it, it closes it up, and it's fine. So the bottle sampling procedure, the Niskin bottle sampling procedure, let's see. So you deploy the Niskin very similarly to how you deploy the CTD. You attach the Niskin bottle to either a line to go down or to a rosette, which is a bunch of Niskins, and then you lower it down to your desired depth, and then you trigger it. Then you trigger the bottles to collect the water. And then you bring the bottles back up to the surface, retrieve the water samples for analysis, and then you conduct lab tests on all the collected water samples. Now, a lot of the times the CTD and the Niskin are used in combination with each other. So combining the CTD and the Niskin bottle sampling, you have a rosette sampler, which usually looks like a bunch of Niskin bottles around in a circle with a CTD device somewhere in the middle. And that allows for simultaneous profiling and discrete sampling. And that's good for data correlation, so you can profile data. It's for different depths, correlation of the depth with different water samples, really. This is really good because it increases efficiency. You can get a whole lot of data with one process. So it's good for efficiency and also just the accuracy of oceanographic studies because you can attach GPS data to it and all kinds of things. So we use this a lot for climate studies. We use this a lot for marine ecosystem studies, studying the ocean temperatures, salinity and currents. We can study nutrient distribution. We can study phytoplankton populations. We can do pollution monitoring to detect pollutants and track the spread in the marine environments. The CTD and the Niskin are two really fundamental techniques in oceanography, and they provide complementary data for really comprehensive understanding. It's really cool the way they do it. There's lots of engineering that goes on that not all of the time I really understand, but yeah, that's CTD and Niskin bottle sampling. If you have any questions, let me know. Thanks.